Welcome to the BioStock studio and today's presentation, which will come from Elicera Therapeutics and CEO Jamal El-Mosleh. Welcome, Jamal, and please go ahead.
Thank you. So Elicera Therapeutics is a cell and gene therapy company focusing on immune oncology, meaning that we develop therapies that uses the patient's own immune system to fight cancer. It's based on many years of research by Professor Magnus Sand and his research group at Uppsala University here in Sweden. And they've been very successful throughout the years to secure different types of research grants, which has enabled us to develop our different drug candidates to quite advanced stages without having to turn to investors, which we did the first time last year. So we conducted an oversubscribed share issue in 2020, and we're currently planning to list the share on Nasdaq First North growth market. In connection to that, we have an ongoing share issue where we're offering shares to the amount of 52 million Swedish kronor. The subscription period has started and will end on June 8th. And I will talk a little bit more about this process at the end of this presentation. We have a broad pipeline of different drug candidates. So we're developing 2 CAR T cells and 2 oncolytic viruses. We have one ongoing phase 1/2 study in neuroendocrine tumors with an oncolytic virus. And we also have a fully developed technology platform, which we call iTank, which can be used to optimize both CAR T cells and oncolytic viruses. And we, of course, use this technology to optimize our own products, but it can also be used to strengthen and optimize the products of other companies' drug candidates. So it's something that we think should be interesting for other players in this field. We gene modify our drug candidates so that they elicit a second mode of action through activation of CD8 positive, so to speak, killer T cells, meaning that we can give rise to multiple mode of actions in one therapy and multiple attacks on cancer through one drug candidate. We have clinical experience from both CAR T cells and oncolytic viruses. Professor Magnus Sand was actually part of the first CAR T cell study in the whole of Europe. This, of course, brings valuable and important experiences, which minimizes the risk that we won't be able to conduct the planned clinical trials in an optimal way. Myself, I have over 13 years of experience in the life science industry. I've been running different types of biotech companies throughout these years, and I've been acting co-founder and CEO of another immune oncology company for almost 10 years. That company is called Immunicum. It's currently listed on small cap, and we IPO'd that company in 2013. We also have a board with broad and complementary competencies, which I will go through shortly. A little bit about the company history. You see on the picture here to the far left, Magnus Sand and his research colleague DU, co-founders of Elicera. They developed an oncolytic virus throughout 2007 and 2011 and completed successfully preclinical studies and basically took the project to the stage where it was ready to enter patient studies. But unfortunately, they lacked the funding to initiate a clinical trial, so they had to pause the development. Around this time, Magnus was contacted by a British freelance journalist that had a friend that had gotten neuroendocrine tumors and had no treatment alternatives. So this journalist, he took to Google and he looked for promising treatments for his friend, and he stumbled upon Magnus Sand's oncolytic virus and contacted him to see if there was a clinical trial in the plans or ongoing that his friend could participate in. And they started a discussion around the problems that is often relevant, at least here in Sweden, where you have interesting innovations in academia, but they don't always reach the patients, since there's often a lack of funding to initiate clinical trials. So the journalist wrote a large article around this, and it got spread in Financial Times, The Guardian, The Telegraph, and other news channels. And this ultimately led to a large crowdfunding campaign and a large donation from a businessman called Vince Hamilton, who himself had gotten neuroendocrine tumors, but unfortunately passed away before he had the chance to participate in the clinical trial. But his family set up a foundation called the Victory Net Foundation that is funding research in neuroendocrine tumors, and then are also then financing our ongoing clinical study in neuroendocrine tumors. The company Elicera was previously named Virex, and it was formed in 2014 by Magnus and Dee. And they formed Virex basically not to run it as a business, but only to where you could keep the assets, the patent applications and the patents around their technologies. I met Magnus and Dee in 2014 when I was CEO of Immunicum, and Immunicum at that point bought a patent from Magnus and Dee. And that patent was regarding a vector that Immunicum wanted to use for one application area, but that it could also be used to develop this oncolytic virus. And of course, Magnus and Dee were still interested in that field, so Immunicum gave Magnus and Dee an exclusive global license to continue to use that technology for development of oncolytic viruses for treatment of neuroendocrine tumors. The study was initiated in 2016, the same year that we also applied for a patent application for the iTank technology platform. Throughout 2018 and 19, Magnus and his group were very successful in generating large research grants, among others from the Swedish Cancer Society, to produce CAR T cells for an upcoming clinical study. I've kept in touch with Magnus and Dee throughout the years, and when I left Immunicum by the turn of 2016-17, we had a discussion regarding the opportunity to collaborate, and we concluded that we for sure had a lot of interesting projects, and we wanted to do something together. But we also concluded that they had sufficient grants to continue developing them efficiently in academia for a couple of years. So we took up discussions again in 2019 and in 2020, and we decided to launch Elicera as a business that for the first time then lifted out, so to speak, the projects from academia and into commercial development. So in 2020, we changed name from Virex to Elicera. We recruited a board of directors, and we conducted this oversubscribed share issue, and we also applied for a patent protection for ELC401, which is our CAR T cell in solid tumors. On this slide here, you see a picture of our product pipeline, and you see we have different projects in different development stages. And the most advanced is ELC100 in neuroendocrine tumors, this oncolytic virus. We also have a second oncolytic virus under development called ELC201, and it's a next generation oncolytic virus in the sense that we have genetically modified this oncolytic virus to elicit 3 mode of actions combined in just one product. ELC301 and ELC401 are our CAR T cells under development. ELC301 is in liquid tumors, where we initially will target B-cell lymphoma. We're planning to initiate a clinical trial starting next year. And 401, we initially target brain tumor, glioblastoma multiforme, but we believe that the target that we're going for is also overexpressed in a number of other solid tumors. So we see a possibility outside of brain tumors for 401. And then, of course, the ELC001 or the iTank platform, it's fully developed. And as I mentioned before, it can be used to optimize not only our own products, but also the products of other companies in this field. So what is an oncolytic virus? An oncolytic virus is a virus that you have genetically modified so that it selectively infiltrates and replicates only in tumor cells, but not in healthy cells. When it replicates in a tumor cell, it does so enough that the tumor cell bursts and dies, and you get something that's called an oncolysis. And this process in itself is something that kickstarts the immune system and activates the patient's own killer T cells that, in parallel to virus particles continuing to attack other cancer cells, you also have a parallel attack of these killer T cells on cancer. So you have you can say a 2 mode of actions in one product here. What differentiates ELC100 from 201 is that ELC-100 is restricted for treatment only in neuroendocrine tumors, while ELC-201 is applicable for treatment of most tumors. ELC-100 is also giving rise to 2 mode of actions, as I just previously here described, while we have genetically modified 201 to elicit 3 mode of actions in one product. The clinical study that is ongoing was initiated in 2016, and you see a picture of the first patient being treated here, and below that you see a picture of Vince Hamilton, who donated a large sum of money for this clinical trial to be able to be initiated. We've also named this product after him, so ELC-100, the oncolytic virus, is called Advince in his name. The study that is ongoing is conducted in 2 steps, so step 1 and step 2. Step 1 is a so-called dose escalation study where we treat 12 patients in different doses with the aim of finding maximum tolerable dose to then be tested in step 2 in an additional 12 patients. And this study is conducted in collaboration with Uppsala University, who are sponsors, and they've also reported promising efficacy data already for one of the patients here, which we think is very encouraging. So going in and describing CAR T cells, what is a CAR T cell? Well, a CAR T cell was named Advance of the Year by ASCO in 2018, ASCO being one of the largest cancer organizations in the world. And the reason that they named CAR T cell therapy Advance of the Year was because it turned out that this type of therapy had the capability to completely eradicate all tumor cells in previously very difficult to treat patients with different types of blood cancer that basically had no other treatment alternatives and were more or less dying. So a CAR T cell therapy is usually you take blood from the patient, you filter out the T cells, you genetically modify them so that they express a chimeric antigen receptor, a CAR molecule. And this CAR molecule then recognizes certain targets on the tumor cell. And then these CAR T cells are expanded in vitro and then re-injected into the patient with the goal of then finding all T cells that carry these targets. They bind to them and then they start killing them. And as I mentioned, this works very, very well in blood cancer, but there's still a lot of challenges in solid tumors. And the reason for this is that a solid tumor has a very hostile immunosuppressive tumor microenvironment that suppresses the activity of the T cell. In addition, it's important that the CAR T cell has a good target on the tumor cell in order for there to be a good anti-cancer effect, of course. And there's very good targets in blood cancer. For example, CD19, which is the majority of CAR T cells under development, target CD19. But in solid tumors, there's a difficulty in finding relevant targets that are not also expressed on healthy cells. So basically, CAR T cells need to be able to survive in the hostile tumor microenvironment and also trigger an immune response that can attack CAR antigen negative tumor cells. And this is precisely what we believe we can do with our CAR T cells under development and what we believe we can help to achieve with our iTank technology platform, which can then be applied to any CAR T cell under development. So to further put this into perspective, we can say that the CAR T cells have been developed in different generations and have been continuously improved throughout the years. So first generation CAR T cells, it turned out that they had insufficient proliferation, they had a brief T cell survival, insufficient immune activation, and for this reason, you optimize them in second generation, third generation, and now also fourth generation CAR T cells. And they've continuously gotten better and better. And what differs fourth generation CAR T cells from other generations is that fourth generation CAR T cells, they contain a cytokine transgene that codes for a cytokine that triggers the immune response in some way or another, usually by activating CD8 positive T cells. So you get a second mode of action. And what differs Elicera's fourth generation CAR T cells for other fourth generation CAR T cells is that we also have a transgene, but we don't code for a cytokine. We code for a neutrophil activating protein from Helicobacter pylori, is short here NAP. And when NAP is activated, it starts a process that releases a whole set of different types of cytokines and chemokines that we believe give a much broader and more optimal immune activation compared to other fourth generation CAR T cells that only use one cytokine. Also, most CAR T cells under development today, as much as 70%, still belong to second generation CAR T cells. And there's only about 16% under development today that belong to fourth generation. And of these, we believe that most of them are being developed in academia. We've actually only identified one other company, a Japanese company that developed fourth generation CAR T cells. And they have a similar technology platform, similar to iTank. They call it PRIME-T. And this company has been very successful in generating multiple licensing and collaboration agreements around their technology platform. And this is something that we aim to do also for the iTank platform. So without going into the specific mode of action of iTank, we can just conclude here that CAR T cells have the ability to target tumor cells through a specific target on the surface of the tumor cell. This is where the CAR molecule binds to. And usually the CAR T cell, they only targets one or maybe two targets on the tumor cell. But of course, the tumor cell contains many more targets, and many more are relevant. And what we can do with the iTank platform is to generate a second mode of action through activation of CD8 positive T cells, the patient's killer T cells, against the whole set of different relevant tumor antigens on the tumor cell, the different tumor targets. So this should give a broader attack on cancer cells, a better capability to withstand the immunosuppressive microenvironment in the tumor, and also an immune response that remains even if the tumor loses the target antigen for the CAR T cell when the patient relapses. So in blood cancer, as I mentioned, this works very well with the CAR T cell treatment. But still, a lot of these patients are treated with blood cancer with CAR T cells. Many of them relapse, and when they relapse, the tumor often loses then the target antigen. And in that case, makes no use to continue treating it with a CAR T cell. So if you have this iTank platform, then you have also a parallel attack with the CD8 positive T cells that can continue to attack the tumor, even if the tumor loses the target for the CAR T cell. We have tested this quite extensively in different preclinical studies, and we have submitted the extensive data for publication. We expect it to be published later this year. So we have highlighted just one study here, in a mouse study where we injected mice with 50% CD19 negative tumor cells and 50% of the tumor cells being CD19 positive. And then we treated these mice with conventional CD19 CAR T cells, and then CD19 CAR T cells that have been optimized with the iTank platform, or with NAP, as it says here in the figure. And we can see that we have a substantial reduction in tumor size and prolongation of the survival of these mice when they are treated with the iTank enforced CAR T cells compared to conventional ones. So this gives you a nice indication that we can elicit this second mode of action through the killer T cells. What we also did was that we extracted blood from these mice, and we tested the reactivity of the T cells against CD19 negative tumor cells. And we saw that only those mice that were treated with the iTank CAR T cells had T cells that could target CD19 negative tumor cells, which then indicates very clearly that you get this second mode of action generated. There's a large interest in CAR T cells, but the field is still relatively young. And what we mean with this is that the majority of the products under development in this field are still earlier than phase 2. So only 5% are in phase 2 or later in development stage. There are 5 approved CAR T cells in different types of liquid tumors, but none have so far been approved in any solid tumor due to the challenges I mentioned before. And this is something that we believe that at Elicera that we can change. Elicera is one of very few companies in Europe. So there are around 100 companies developing CAR T cells throughout the world. It's a roughly 50-50 split between US and China, a little bit more companies in China. Only 14 companies developing CAR T cells in Europe and only one in Sweden, being Elicera. Most of the, half of the CAR T cells under development are targeting CD19 still. In China, as much as 85%. So it's still a lot of focus on blood cancer due to the challenges that there are seen in solid tumors. So ELC-301, again, is our CAR T cell targeting blood cancer, where we initially go for B-cell lymphoma. This is an indication where we already today have 3 approved CAR T cells against CD19. So you can ask yourselves, why would we go for this specific indication if there's already approved CAR T cells here and they work so well? Well, they do work very well, but according to recent studies published in Nature Medicine, where they have looked at how the patients respond to these treatments, there's 40% that have a sustained complete response. And this is, of course, amazing results. It's very good, but this also means that the majority of patients are still in need of additional treatments, even though you have these CD19 approved CAR T cells. And ELC-301 targets CD20, not CD19, and we know that CD20 is also overexpressed on B-cell lymphoma. So for that reason alone, it can be used either before or after treatment with conventional CD19 CAR T cells. But also, as you remember, we've enforced this with the ITANK technology platform. So we believe that we can also generate the patient's own killer T cells to target also other antigens on the tumor besides CD20 and CD19. We have an ongoing GMP production for ELC-301. And we plan to initiate the first clinical study with our CAR T cell, this 301, in the second half of next year. The business model is based on out-licensing. So our ambition is not to take a product all the way to the market, but rather reach proof of concept in clinical studies, minimize the risk and add value to the projects, and then find a partner that can take over development and complete the late-stage clinical trials and also market the products. And we see multiple opportunities, as I said. For the CAR T cell field, of course, the ITANK platform is part of both of our products under development. But we believe that the ITANK platform can be out-licensed on its own. And it's a platform that we believe is highly interesting for any CAR T cell player out there. ELC-301 is a little bit more niched in a few blood cancer indications, while ELC-401 initially will target brain tumor. But we believe that the target that we're going for, IL-13RA2, is also overexpressed on a number of other solid tumors. So we see multiple opportunities for out-licensing for this product. Looking at the oncolytic virus field, again, we have a more niche product in ELC-100, specifically restricted to neuroendocrine tumors, but ELC-201 is applicable against most cancer indications. So here we also see multiple opportunities for out-licensing. A short description of the team. You can say that I'm an educated biotech entrepreneur. I have a Master of Science degree in Industrial Engineering and Management with a focus on biotech, and a master from Chalmers School of Entrepreneurship, focus on life science. 13 years of experience from running life science companies, and 10 years as CEO of immuno-oncology company Immunicum. Ingvar Carlsson is our CFO. He has 20 years of experience as CFO for both public and private companies, among others as CFO for cell therapy company Idogen. And Magnus Sand, Chief Scientific Officer and co-founder, professor in gene therapy at Uppsala University, previously employed at National Cancer Institute. He has received multiple awards and has multiple publications and clinical experience from both CAR T cells and oncolytic viruses. And me and Magnus are two of the larger shareholders of the company. The board of directors, besides these persons that you see here on this slide, also consist of Magnus, just to mention that. And Agneta Edberg, being our chairman of Elicera, was also my chairman at the Immunicum. So we have an ongoing collaboration, and we know each other since many years back. And this is, of course, something that gives us a very good ground for collaboration. She has a broad business strategy profile, over 20 years of experience from the life science industry. And she knows immuno-oncology and cell therapy. Kristina Heder, similar profile, broad business strategy profile, plus 20 years in the industry. She's today the COO of another oncology company listed on Nasdaq called Medivir, and experience from several board positions in public companies. Jan Zetterberg is our legal expert with many years of experience from senior positions within AstraZeneca's legal department. Margit Jorvid, our regulatory expert, plus 30 years in regulatory affairs. She was previously head of regulatory affairs and QA at Immunicum. She knows cell therapies, she knows immuno-oncology, and her experience is very valuable for us. As is Karin Hågendorn, recent addition to the board. She's our CMC specialist with a focus on cell therapies and immuno-oncology, and she also participated in bringing Kymriah CAR T-cell to the market at Novartis. Her experience is, of course, very, very valuable for Elicera as well. So short on our clinical development plan for the coming 3 years, you see an overview here, and you see that we have an ongoing phase 1/2 study in neuroendocrine tumors, where we plan to report step 1 by end of 2022. Next year, we also intend to start our first clinical trial with ELC-301 in B-cell lymphoma. And from 2023 and onwards, we expect to have quite a broad clinical program with the different clinical studies ending then in different stages. So we're looking for 52 million kronor now before share issue costs, and this should last us a bit into 2023. So the share issue in brief, it's 6.5 million units, and 1 unit is 1 share plus 1 warrant. We have priced this at 8 kronor per unit, meaning that we're looking for 52 million in total. We have an over-allotment option of up to 10 million kronor if the share issue is oversubscribed. And the exercise price is set at 1160 Swedish kronor for 1 new share with the support of 2 warrants during the period of November in 2022. Elicera today has a pre-money valuation of 96 million Swedish kronor, and we have subscription commitments of 64% of the offer, 33 million, of which 9.3 million comes from board management and larger shareholders. Subscription period between May 24th, so it's ongoing, ends June 8th. And you can see more information on this and all the relevant documents and how to subscribe and so on our website, elicera.com. So to end up here and this presentation with a short summary, it's a newly launched immuno-oncology company in the field of cell and gene therapy. Substantial research grant throughout the years has enabled us to advance the projects quite far. We turned to investors first time last year, and we're planning an IPO now, shortly. We have a broad product portfolio of CAR T cells in both solid and liquid tumors, next generation oncolytic viruses, ongoing phase 1/2 study, fully developed ITANK platform, unique positioning with our 4th generation CAR T cells, unique positioning with our 3 mode of action in 1 oncolytic virus in 201. And we have a team with relevant experience, clinical experience from CAR T cells and oncolytic viruses, management with experience from immuno-oncology, and a board with very complementary and relevant experiences in different areas. And with that, I thank you so much and open up for questions.
Thank you too, Jamal, and we have a couple of questions for you. First of all, immune oncology is undoubtedly a really hot topic in the life science industry at the moment. What is it, in short, that makes your technique stand out from the others?
So immuno-oncology is a broad term in the sense that there are many different types of therapies that go under the term immuno-oncology. So what makes us unique is that, as I said, we're developing CAR T cells and oncolytic viruses, and depending on how you look at it, it's either very unique or somewhat unique. So it's we're not unique in the sense that we're not the only ones that develop oncolytic viruses and CAR T cells, but we are unique in the sense that we develop a very special type of drug candidates within these fields. So a fourth generation CAR T cells, as I explained before, it's quite unusual that you find those under development outside of academia. So still most of the CAR T cells under development are still second generation. So we're unique in that sense. And from a geographical perspective, in Europe, as I said, it's also very quite unique just to have developed these types of products.
And you did touch on the market and the market potential a bit in your presentation, but just to recap, could you tell us about the market potential for Elicera?
Yeah, I mean, obviously these products, if they reach the market, there's a huge potential for them. So I can say that it's different market potential for different drug candidates in our portfolio. We have CAR T cells and oncolytic viruses that are both more niched and more broad. So ELC-100, the oncolytic virus in neuroendocrine tumors, of course, has a smaller market potential, being that it's restricted to that specific indication compared to ELC-201, where we see a much broader market potential since it can be applied to most cancers under development. And it's a little bit the same with the CAR T cells. So ELC-301, it's more niched against blood cancer, while 401 we believe can be used to target a range of solid tumors, and there the market potential is bigger. And on this question, I also want to mention again the iTank technology platform, where we believe that we can generate interest from multiple companies in these fields around that technology platform alone. So there we also see a huge market potential for that platform.
And like you mentioned, you are carrying out a share issue. And could you tell us a bit about how you plan to spend the money that you raise?
Yeah, so we have an ongoing phase 1/2 study, and some of the funding will go to continue to conduct that clinical study as efficiently as possible. And a lot of the other activities, of course, are around advancing the other drug candidates under development towards clinical trials as fast and as efficiently as possible. So the next one to enter clinical trials is 301. ELC-301 in B-cell lymphoma. But we also intend to advance 201 and 401 as soon as possible into clinical trials. So parts of the funding there will also be to complete preclinical studies and also initiate GMP production, which is costly and takes a lot of time. But since we want to move to clinical trials as soon as possible, we want to start with these activities also as soon as possible.
And just to finish off then, when we end 2021, what achievements do you hope to look back on?
I hope that we can look back at a very successful IPO here, of course, being that it's currently the focus of the company, and conclude the share issue, also the funding, in a successful way, which I believe that we will. And then, of course, to have initiated the different activities that we want to initiate here during the fall, so that we can move our drug candidates to patients as soon and as efficiently as possible. So I hope to be able to look back on 2021 and see it as the year when we laid the foundation for a broad clinical program the coming years here, where we treat patients successfully with our drug candidates.
Well, thank you so much for joining us.
Thank you so much for the opportunity.